For multiple sequence alignment, we are now studying what are different algorithms for multiple sequence alignment. So basically, we will look into two algorithms, which is number one is extension of dynamic programming, which is called exhaustive search, and the other one is progressive alignment techniques. So for exhaustive search, the first method is sum of pairs method, which is basically a dynamic programming based method. And what we are doing, instead of comparing only two sequences, we compare more than two sequences, and then we find out the scores for individual sequences and then sum the complete score. So what happens is that we compute pairwise score or of all the pairs of characters, and then we sum them, and then we find out aligned columns which are common in all the sequences. So if I have four characters which are shown by I minus I and V5 sign, so what will happen? We will have six total comparisons which are shown by Si then dot plus Sii and so on. So for four characters we have six comparisons. So as it can be seen that with the increasing number of characters, it would be very extensive computational alignment. So what is the solution? Again, before going into solution, I will see that what is the problem basically. So for 2D matrix, what we are doing, we are converting into n-dimensional matrix. So firstly, we have two sequences for sequence alignment. For MSA, we have n sequences. So it would be n-dimensional matrix, and complexity is of course increasing with the number of sequences. For example, if we have roughly less than 10 sequences, we will at least need more than 200 comparisons to align those characters. So for that purpose, we will move on to the other technique, which is progressive MSA. So progressive MSA क्या होती है? Progressive MSA के three steps हैं. Number one step is again do pairwise sequence alignment using some dynamic programming or global alignment approach. The other one was create a guide tree which would be based on some distance finding algorithm such as neighbor join, which we will see in a moment. And the third one is once we have got the guide tree, then we try to align sequences based on the distances from that guide tree. आइए इन स्टेप्स को हम तफसीलन ज़रा देखते हैं कि वो स्टेप्स कैसे होंगे. फॉर एग्जांपल, फर्स्ट स्टेप जो है वो हमारा सिंपल सीक्वेंसेस को हमने अलाइन करना है यूजिंग कॉमन डायनेमिक प्रोग्रामिंग टेक्निक्स. अगर हमारे पास टोटल n सीक्वेंसेस हों तो इसके लिए हमें फार्मूला चाहिए होगा n-1 into n by 2. So for example, if we have 5 sequences, then we would at least need 10 comparisons. If we have 4 sequences, then it would be 4 into 3 by 2, it would be roughly 6 comparisons, and so on. So first step is to find out common comparisons between the given sequences, and again it will increase with the number of sequences. So second step is construction of guide tree. So basically, what is a guide tree? It is basically a distance based measure. So how it is used? It converts similarity scores to some distance scores, and then use some clustering algorithm to put those sequences which are have very slow score or low score into common clusters. And then a tree shows the distance between objects. And finally, this tree, it is not a phylogenetic tree, which is different from a guide tree. So what is the stage 3? Stage 3 is after we have got the guide tree. Now we want to assign our sequences to their following guide tree. So what we do, we make successive alignment based on order in the guide tree. That is, for example, we will try to find out which are the closest sequences in the guide tree, and then we will compare them. Then we will take the second column, second closest one, then third close, and so on. Then what is the rule? Basically, rule is that if we have a gap, then it will always be a gap. Why? Because if we have a gap in the start, and if we want to remove that gap later on, it's not possible, because it will be accumulated, or it will be increasing by in each step. So we can't remove the gap once it is delivered there. So what are the problems with MSA? So as you can see, for progressive MSA, there are common problems such as in initial step, we are comparing or we are putting multiple sequences using dynamic programming techniques. So if that number is very high, it will be very slow, and it will be very computational expensive. And then finally, errors in initial alignments are propagated, so that cannot be removed on later on stages. So similarly, gaps can be reflate. That gaps keep on increasing with each comparison, and they cannot be removed at later stages. And finally, there can be some gaps which are based on amino acid specific, and we see that these are gaps, but although they are conveying good information, so that information is also lost by in PMSA techniques.